Welcome everybody from IST Austria, the Institute of Science and Technology Austria. My name is Tom Hensinger. I welcome everybody in the IST community, and I also especially welcome all our guests today, our guests in Austria and abroad. The IST Lecture series is our most prestigious lecture series, and this is our first remote IST lecture. Given the times we are in, we will be moving many of our formats online, also other lecture series, colloquia, seminar series. And while we, of course, would love to have the speakers on campus to talk with them in person, there's one advantage to these remote lectures, namely we can have much broader participation so I'm very much looking forward to today's talk, and I'll hand over to Leonid Sazonov to introduce today's speaker.
Thank you, Tom. Welcome, everybody. So I'm Leonid Sazonov, professor in structural biology here at IST, and I'm especially happy to see Joachim today as our guest, because we have several groups working in cryo-EM in IST and only recently installed high-end electron microscopy suite here. So this is very timely. And of course, it's also timely because incredible developments in cryo-EM, which happened in the last few years, in large part thanks to Joachim efforts. So short biography, Joachim Frank is a professor at Columbia University in New York. He received his PhD from the Technical University in Munich, and in 1975 he joined the Wadsworth Center in Albany as senior research scientist, and then in 1985 he joined faculty at State University, New York, Albany. And in 2008, he moved to New York to his current positions. So Joachim Frank Lab is very well known, of course, for his developments of techniques, especially in software, of single particle reconstruction, of macromolecules, and he applied these techniques to visualize the structure and dynamics of ribosome during protein synthesis, and he now develops this to time results studies and conformational studies and so on. So remaining at the forefront of the cryo-EM development. Professor Frank has many awards. He is a member of National Academy of Sciences in US and American Academy of Microbiology. He is fellow of American Academy of Arts and Sciences and the American Association for Advancement of Science. And he was, among his awards is Franklin Medal for Life Science in 2014. In 2017, he shared the Value Prize in Biomedical Sciences with Richard Henderson and Marin Van Hill. And of course, he was awarded 2017 Nobel Prize in Chemistry together with Jacques Dubochet and Richard Henderson. So we are looking forward to the talk. I'm handing over, please. Please, Joachim.
So I would like to thank the organizers for inviting me to give this talk. At the time of the invitation, the world was still a familiar place. Yesterday, as it was planned originally, my wife and I were due to arrive by Air France in Vienna. But it is gratifying that in isolation, we still find each other, that we are still able to connect as a global audience to discuss science and the challenges we face. So today, I'd like to talk about single particle cryo-EM, visualization of biological molecules in their native states. So I'm showing the animal cell. We want to understand the interactions between molecules in the cell. In terms of the whole program of structural biology, we want to understand not just the structure, but the way molecules interact in supporting all life functions. Here's a picture of a cartoon of the animal cell. And it is, I'm showing it to give you an idea about how closely packed everything is. If you were to understand the structure and molecular interactions on the atomic scale, it would be very, very difficult to do this with a packed environment like this. And so we are, what we do is we exercise reductionism. We essentially, we take molecules and their interacting partners out of the context of the cell into an in vitro system. Now, this is a gross simplification, but it is the only way how we can do this. In many cases, it is actually possible to look at a molecular machine in vitro by providing all components and all energy supplying entities. So we can essentially follow in a very particular process outside of the context of the cell. So reductionism is really underlying our entire approach. And we defer the question to what extent what we find is actually representing what is happening in the packed environment. We defer this. We have to essentially approach this question using other techniques. And associated with the concept of the molecular cell, of molecular machines, is Bruce Alberts. He made this this idea popular in 1998 in a very important article in Cell. And so he essentially, what he was saying is we have underestimated the cell. It was used to be seen as sort of a sac with interacting molecules, but in fact, it is very highly organized. And on the first level of organization is the molecular machine. A molecule interacts with several ligands, and very often in a processive way, which means that there is a before and an after. There is a time in molecular dimensions, and that the direction of time actually arises from the expenditure of energy. So there is always an event which marks the division between past and future. Here are a number of systems, molecular machines that we have become familiar with from various studies, including structural biology. So ATP synthase makes ATP, proteasome recycles proteins, RNA polymerase copies DNA into mRNA, ribosome makes proteins, chaperone folds proteins, the dynein transports molecules, spliceosome edits mRNA in eukaryotes, and the flagellar motor rotates flagella. Here's an example of multiple states of a molecular machine, the ribosome. This is an illustration that was put together by Venki Ramakrishnan. And it shows that there are many states that follow in sequence, and some of them are cyclic. The elongation is a cyclic process. This gives you an idea about how gigantic the program of structural biology is, because we want to characterize all the states that we see in this flow diagram, but we also want to understand the intermediate states that are not even shown here. So in my talk, I'm going to talk about the ancient history, EM and X-ray crystallography, then the single particle techniques, the concept, and then the first single particle reconstruction, and then cryo-EM reconstructions once cryo was invented, with increasing resolutions up to 5 angstroms. And from 2012, which is a milestone event, to now, we have a resolution revolution, which we still experience. And today, there's an exponential increase in cryo-EM structure depositions. And what is new are a number of opportunities, such as time-resolved cryo-EM and the mapping of continuum of states using cryo-EM. And I will only talk about this very shortly. So it might not be appreciated how close the interaction between cryo-EM, between electron microscopy and X-ray crystallography was at the very beginning. Here I show one of the first what we would call hybrid meetings today, was in Hirschegg, which was the site of a 1968 workshop on X-ray crystallography and EM of proteins, organized by Walter Hoppe and Max Perutz. And among the participants, whose name, of course, ring a bell today, is Harold Erickson, Richard Henderson, Ken Holmes, Hugh Huxley, Nigel Unwin, and many others. So this was really, since I started my PhD thesis under Walter Hoppe in 1967, this was one of the first events that brought me together with people who became leaders in their respective fields later on. Here are snapshots from the conference site of the Hirschegg meetings. And here we have Walter Hoppe with Max Perutz and Hirschegg. So both interacted because Walter Hoppe was an X-ray crystallographer initially, who developed later on an interest in electron microscopy. But he co-organized these meetings and workshops together with Max Perutz, which started initially as workshops in protein crystallography and then became all-embracing and embraced the people in electron microscopy. Now I have to, since I have a relatively large audience, I'd like to tell you about some basic concepts about X-ray crystallography and electron microscopy. So X-ray crystallography deals with crystals that are very highly regular arrangements of molecules in regular order. So in a crystal, we have many copies of the molecule. And with the exposure to an X-ray beam, we get a diffraction pattern. And with techniques called phasing, we can determine the structure. And what the picture shows here is Max Perutz with his student John Kendrew looking at the structure of hemoglobin in 1962. And this was the first, one of the first protein structures that were obtained with X-ray crystallography. Now, for proteins which have a composition of very large number of atoms, the X-ray beam must be high intensity, the crystal must be almost perfect. And to date, and this has to be appreciated in the context of cryo-EM, to date 140,000 structures that were solved by X-ray crystallography are now available in the public database. So these are 140,000 structures of proteins that are all relevant to human health. Now, there are problems with X-ray crystallography because of the crystal packing. The molecules are not visualized in all conformations and binding states that are important for function. And sometimes the structure has no relevance at all. And then what is even more important, many molecules do not form highly ordered crystals. So those have been sort of outside of the radar. They could not be studied at atomic resolution. The shortcomings, many molecules do not form highly ordered crystals, and they are sort of outside of the radar of structural biology. The sample quantity can be a big issue as well. And electron microscopy can be used to solve molecular structures as well, as you all know now. Projection images are formed at very high magnification. A typical number is 30,000. And to reconstruction object, many different views must be collected. And there are some shortcomings too. The sample must be very thin because electrons are readily absorbed by matter. And also the electrons strongly damage the molecules. There's a need for low dose. 10 to 20 electrons per square angstroms are the typical number. But the result of this is that the images are very noisy. They are affected by the so-called shot noise at low exposure, which simply means that it is impossible to interpret molecules just from such an image. One needs to use averaging in a massive way. So one of the effective ways of averaging is, of course, the averaging over molecules that are formed, that are forming a crystal. So again, we are back to crystals here. In the beginning of molecular electron microscopy, there have been several pioneers that applied similar methods as X-ray crystallography to molecules that are arranged in very accurate order. And the pioneers in electron microscopy were Aaron Klug and David Dorosier. They used for the first time a mathematical theorem that was developed by John Radon in Germany in 1917. So obviously for decades, this theorem could not be used. And this year was the first application of this theorem. And you see essentially a view of this on the right-hand side. The idea is to collect multiple views, and each views, when you Fourier transform it, is actually a section, a central section in the three-dimensional Fourier transform. So the recipe for three-dimensional reconstruction is simply that you collect many, many views in a large angular range and then perform two-dimensional Fourier transforms and arrange these in the three-dimensional Fourier space, and then perform some kind of an interpolation. And so you retrieve the information in three-dimensional Fourier transform, and then all you have to do is go back into real space by a three-dimensional inverse transform. So that's in a nutshell is a recipe for three-dimensional reconstruction. Now, Aaron Klug and David Rossier applied this first to bacteriophage tail, which is highly ordered in a helical way. So they develop programs that make that happen. And in fact, for helical structures, you only need a single view in order to create all the other ones because of this particular symmetry. Now, other people also approached highly ordered structures. Tony Crowther in 1970 obtained three-dimensional reconstruction of viruses with icosahedral symmetry. Here you have 60 units in a perfect icosahedral order. And so a single view already gives you a lot of information, and you might only need 2 or 3 in order to reconstruct the entire particle. So now you get the idea. The programs and the computational procedures that needed to be used were tailored to a particular geometry. So in the first case, it was helical symmetry. Here it was icosahedral symmetry. And here's another example, a planar two-dimensional ordering of, of molecules. This was the work of Richard Henderson and Nigel Unwin in 1975, and it was it was a milestone in electron microscopy. They were the first to get really high resolution, 7 angstroms for the 7-helix bundle of Bacteriorhodopsin. They also used, instead of using negative staining technique, they used glucose embedding, and that was a big step forward. And they were also the first ones to use very low dose, so they were sure that the molecule was not being damaged. But this is a third example where programs and the mathematical techniques were particularly used, developed for one particular symmetry. And so there was no general solution to the problem of reconstructing a molecule that was not in crystal forms. So one of the people who asked the question, why crystals? Couldn't we simply use the molecules as they are and put them on a grid and then do what's called electron tomography? In electron tomography, you tilt the EM grid into many different angles, and then use the radon theorem, the projection theorem, in order to fill the Fourier space, and then do the inverse Fourier transform. So the idea was simply you have the molecule sitting on the grid, and now you perform electron tomography. So you essentially reconstruct the grid along with the molecules, and then you can simply pick the three-dimensional reconstructed molecules and look at them. You can now also average them in 3D. So that was the idea. But the problem with this approach is that the accumulated electron exposure exceeded 1,000 electrons per angstrom square. So that was not realistic because it burned the molecules essentially to cinder, and the three-dimensional reconstruction did not have any meaning. So I asked the same question, why crystals? And so my approach was entirely different. It was the idea to make use of the fact that in solution, the molecules are completely randomly arranged. And so if you take a snapshot of these, then you get with high probability the entire angular range of randomly oriented views. And then so the information comes from images of single, which means unattached molecules that exist in many copies. And the molecules are free to assume all naturally occurring conformations. Molecules are randomly oriented, so a single snapshot may already give us hundreds of particle views. And of course, hundreds of particle views are not sufficient. You just keep collecting and gradually fill the entire angular range. So that was the idea. And I knew from my PhD thesis that images can be aligned to within beta of 3 angstroms. And among the different subjects I studied in my PhD dissertation was the use of cross-correlation functions to do image alignment. And by the way, my dissertation at the TU Munich was published in 2019, last year, 49 years after completion. And I'm very proud that something that I did 50 years ago has still relevance today. So in 1975, I published an article in ultramicroscopy, averaging of low exposure electron micrographs of non-periodic objects. So that made reference to Unwin and Henderson's technique because they were able to get the 7 angstrom resolution. And my article essentially posed the question, what if we were able to do the same with molecules that are not in crystal order? And so it was a purely conceptual article that made sure that people understand that we have the tools available to do this, and all we need is really to develop the necessary programs. So around the time when I was at the Cavendish lab in Cambridge, I met Owen Sexton, and together we studied the conditions for alignment of 2 images of a molecule of size d in terms of particle size, contrast, resolution, and critical electron dose. So the question was simply, is it realistic to apply these ideas that I had in 1975 to the EM images of low dose images of molecules? And so the answer that we found out by investigating this was encouraging. It was really, the solution was an inequality between the particle size and the contrast, the resolutions that we aimed for, and the critical dose. And we determined that for a molecule of the size of the ribosome, and the contrast that we would get for pure ice, which didn't exist at the time as a technique, and a resolution of 3 angstroms, and a reasonable critical electron dose, we were able to really show that, yes, the alignment accuracy would be sufficient to get to 3 angstrom resolution. Okay, so this was for me, a very big motivating event to go further. So now we have something that is supposed to work, but there were no programs available. The devil was in the detail. The problems that needed to be solved was alignment of images. Well, I had a procedure for that. the estimation of resolution of an average so that we have really a way to talk to each other as scientists with different groups. You know, we have to agree of what we accomplish. It's not just a subjective matter. We have to have a tool to sort and classify images because we cannot simply go by the assumption that they're all the same. The biggest problem was finding projection angles and how to reconstruct in 3 dimensions. Well, how to find projection angles is of course the biggest problem, but then reconstruct in 3D is another problem because all the existing programs assumed regular geometries and not random ones. Okay, so we had to put this on the table as well. So faced with these problems, I developed SPIDER, a modular image processing system. So the idea is we cannot simply continue to develop one gigantic all-purpose program. We need a modular program. We need a whole set of what I would call primitive programs and connect them together so that we have a workbench with which we can construct solutions to essentially any problem that we face. So these primitive programs are, you know, how to do autocorrelation, how to do cross-correlation, Fourier transform, rotation, shift, and so forth. So I had an entire collection of 400 of these primitives, and I had a way of stringing them together in a image processing system in which we could use commands and to put together a script language. So that was a sort of a solution to this daunting problem that we were facing. Now, I have to tell you something about the ribosome and its role in the development of single particle techniques. Around the time when I gave talks about the idea of single particle techniques, but I didn't really have a very convincing demonstration, Miloslav Bublik, who was at Rush Institute in Nutley, New Jersey, approached me, you know, can you do something with these here? And he had these beautiful, beautiful images of ribosomes. These were 40S subunits, small subunits of a eukaryotic ribosome, in fact, of HeLa ribosomes. And so they had very distinct features. Now, the ribosome is a very fortuitous molecule to develop this technique with because it has high contrast on account of its phosphorus contents. It is relatively stable, so it doesn't really do a lot of conformational changes, and it has a large size. So these are all very optimum conditions for developing this. So at the time, what did we know about the ribosome? Well, there was Jim Lake. Jim Lake had looked at thousands of images of ribosomes, and he did what I would call tomography in his mind, just by looking at all these different views and also tilting the specimen and observing what he saw. He created three-dimensional models just in a subjective way. And this is the model that, in fact, existed as sort of some kind of a ceramic item with a copyright on it, and he shipped it to a number of people he thought were very important in the field. And so I got one of the copies of this. So in other words, there was really no rational way at the time to get to the ribosome structure. So this is why I adopted the ribosome for development technique. So as a proof of concept of the alignment technique, I used Milas Bublik's 40S ribosome images, and they were left-facing or right-facing, and I selected the left-facing ones, and showed that with these alignment methods, correlation methods, we were able to get reproducible averages. And that was a very big thing at the time, and it was published in Science, and it was, for me, it was a way of getting my first grants from the National Institute of Health. So, but the next big problem is the problem of heterogeneity. The molecules are in different orientations and conformations. And here are examples from the ribosomes that I just showed you that were in left and right phasing views. And then there were the flip and flop views of hemocyanin and the rocking behavior, which is another example. Hemocyanin was very important at the beginning because it was a molecule with which one could really develop these kinds of classification techniques. And together with Marijn van Hiel in 1981, we found a way to sort images using a technique that had been used for blood samples for a very long time, but it had never been applied to images. So it was multivariate analysis of aligned molecules. So with this, we solved this very, very big problem. And from then on, we had an easier time to move forward. Now, one of the biggest problems is how to find the angles of projections. Well, there is so-called bootstrap techniques in which we essentially come up with some kind of a preliminary reconstruction. Once we have the preliminary reconstruction, we can then do some kind of an iterative technique in order to refine this. But the big problem is how to get such a bootstrap reconstruction. So I had a bright idea in 1979, and then that would only got to a result in 1986. So there was very hard work in between program development, and I have to credit Michael Rademacher with this. So here was the bright idea, which I put together in an overhead in 1979. And then later on, American scientists helped me to beautify this in a fancy version. So the idea is that we have, when the molecules are all in the same orientation on the grid, then they have 1 degree of freedom. They can have different azimuths, and but this doesn't give us any new information. But when I tilt such an arrangement of molecules, then the projections are unique. They're unique three-dimensional projections, and from the point of view of the molecule, it looks as if the electron microscopes were walking around in a cone, and then I get all these different conical projections. And we call it random conical because these azimuths here that originate the placement on the cone are, of course, randomly oriented. So that was the idea. And then Michael Rademacher was the one, a postdoc who was also trained by Walter Hoppe, who put it in practice. And he developed a whole suite of programs that were necessary as an sort of an addendum to the SPIDER program, and he made it happen. So here is the first reconstruction of 50S ribosomal subunit from an E. coli ribosome. Now, I have to emphasize that up to that point, everything was negative staining. There was no cryo involved up to this point. So one of the ways how we visualized the 50S subunit at the time was at a contour stack in 3D. So each section of the 3D structure in the computer was represented as a contour, and then we put all the contours together with the right kind of Z distance and mounted them in a wooden box. And when I was asked later on, 2 years ago, by the Nobel Museum to bring something that was sort of idiosyncratic to this entire development or invention, I thought this was really the best thing to bring them. Okay, so now it is on permanent exhibit in the Nobel Museum in Stockholm. So this was a jump forward, obviously, and now I go back into the time. Here we are at the Nobel Museum presenting the various tools, and next to me is Jacques Dubochet, who has a sort of a prototype of the plunge freezer or freeze plunger. And here on the other side is Richard Henderson, who presented the first atomic resolution model of the bacterial rhodopsin, which we obtained in 1989. So this is the big moment when the cryo techniques joined in. And I'm obviously, Jacques Dubochet is one of the important people here, but I also show Bob Glaser, who in 1976 came up with essentially a plunge freezing technique. So he had essentially already the idea, but the difference was that he used liquid nitrogen all by itself. And that poses a problem. When you immerse the grid into liquid nitrogen directly, then air bubbles form on the interface between the sample and the cryogen, and these air bubbles prevent fast heat transfer. Under those conditions, you get the formation of crystalline ice, and that's bad. That crushes the molecule that produces all kinds of damage. So Dubochet's contribution was to use a little container with liquid ethane or propane or a mixture of the two on liquid nitrogen temperature. Under those conditions, the plunging results in almost instantaneous heat transfer. And under those conditions, the water becomes vitreous ice rather than crystalline ice. Okay, so this was really in 1981, but it took a number of years until this whole methodology spread. And we did our first reconstructions in 1988. So the first publication relating to this was in 1990. This is a microscope that was developed later on, but just to show that these were the kinds of cryo-EM, the specialized cryo-EMs machines that soon proliferated. And this was the F30 Polara of FEI. And we got the first of these instruments in the year 2000. when I was still in Albany. So the plunge freezers, here's a manual one, and then there are commercial ones that are available. This one here you can build for $200, and the other ones cost something like between $50,000 and $70,000. These here have the advantage that you have environmental control and you have computer control over the blotting as well as the plunging. Okay, so here we have ribosomes recorded on film. You see the contrast is relatively weak. You have a lot of salt and pepper appearance from the low dose. I just should mention that after the bootstrap reconstruction, we need an iterative angular refinement, and this principle is shown here. So around that time, in the beginning of the 90s, that was very, very enthusiastic situation where we applied this new technique of cryo-EM and single parting techniques to a variety of molecules. And for many of these molecules, this was the first time that they could be seen in 3D because crystals could not be formed. And I'll show you just 3 examples here. And for me, the most interesting was the E. coli ribosome, which for the first time got at a resolution of 25 angstroms, and it showed very interesting and intriguing detail. It was published in 1995 in Nature. At that time, the realization was, well, we can obviously improve resolution, but we didn't have any hope to get anywhere near atomic resolution with this technique. So, but as it was, it gave us information about the changes in conformation, a much more realistic view of molecules than the ones that were confined in crystals, as in X-ray crystallography. So here's the again another view of the E. coli ribosome in 1995, and it gave us an idea on how protein synthesis would work in practice. So this was all modeled, the tRNAs and mRNA and the track of the polypeptide chains were all modeled into it. Around that time, maybe already in the beginning of the 90s, I thought that I could really make contributions, significant contributions to the ribosome structure, as well as the understanding of the progress of translation. And so I hired biochemists as postdocs and who brought in the relevant expertise. So my lab then became one part methods development, and the other part was development of ideas on how the ribosome works. And here we came up with the first elongation cycle. The elongation cycle is the cycle that is used for adding each amino acid in sequence. To arrange this, we had to find the way the relevant ligand molecules bound to the ribosome, and these were the tRNAs, mRNA, or EFG, the elongation factor that moves the ribosome along, and after the peptide transfer, and then also EFTU, which together with the tRNA enters the ribosome in order to satisfy the next decoding event. So the next stage in the development was the maximum likelihood methods of classification. This was all done before the new development with it came from the introduction of the cameras. So with using maximum likelihood methods of classification, which were developed in the lab of Jose Maria Carrazzo by the student Stuart Shears, he showed it was possible to disentangle the orientational and the conformational changes that are visible in projections. So it was possible for the first time to classify the existing structures in a sample and extract several reconstructions all at once. And it was a very, very a surprising development to me that one would be able to do this in this manner. And so I called it story in a sample. We could really apply this in order to extract multiple structures all from the same sample. And this is particularly interesting when you have a molecular machine that exists in many different conformations and ligand binding states. So you can make an experiment. You do not have to actually introduce conformational changes by some kind of chemical intervention. You can simply look at the entire conformation spectrum all at once. It was very exciting to me that we were able to do this. So there were many milestones in single particle reconstruction, and I show this here symbolically as some kind of an avenue. And I'm stopping here in 2013 because this was a very interesting place to wind up, because this was the best resolution that we obtained from recording on film, 5.5 angstroms. An incredible amount of work because these were probably 200,000 images that had to be collected, and they were all recorded on film. So the film scanning, the development, and so forth was such a pain. So, but we got stuck somewhere, and other people got stuck too with an asymmetric structure. Never anything was achieved beyond this kind of resolution, beyond 5.5 angstrom resolution. And then came the very big excitement from the introduction of the single electron detecting cameras. So it was a new recording technology. It was developed independently by 3 different manufacturers who were in contact with academic groups. And these cameras brought an revolutionary increase in signal-to-noise ratio. They were much better than film, and they made it possible, within a couple of years, made it possible to get near atomic resolution structures of proteins and ribonuclear proteins. And here you can see the drastic comparison with the pictures that I showed you before. It shows you the crispness and the higher resolution, higher signal-to-noise ratio of these of ribosomes. And we took advantage of this opportunity, and I'll show you just a few examples of this. Here's elongation factor G mutant bound to the ribosome. This mutant makes elongation factor G ineffective in GTP hydrolysis, so it gets stuck just at the point when it's supposed to trigger GTP hydrolysis. And from this particular structure, we got clues that told us about the way EFG triggers the translocation. And for me, this was really the most fantastic development, that we would be able to get a ribosome structure at 2.5 angstrom resolution. This was in 2018. And you can see in very great detail the details of base pairing, and here are examples for different bases. And we see a protein, a secondary structure, and we can see a water molecule even. So after decades of development, to see a water molecule of a cryo-EM was an incredible and euphoric experience. I just show you this in a little bit more detail. And one point I have to make here is that unlike X-ray crystallography, we have local, the concept of local resolution. The resolution is locally different, and typically in the interior of the molecule, you have high resolution. Here it goes all the way to 2.3 angstroms, and it's indicated by deep blue. And on the periphery, you have less resolution indicated by colors that go into the orange. Okay, here's another example, AMPA receptor. I mentored, co-mentored at TWOMI, who made a number of discoveries of receptor structures. This was here published in Nature. He had a couple of Nature articles in Science. Then I got involved in a collaboration with Wayne Hendrickson and Andy Marks on the structure of the ryanodin receptor. Now you recognize the ryanodin receptor from a slide that I showed earlier, where we only achieved 30 angstrom resolution, and now we get all the way to 3.6 angstroms for different conformation and activation states. So the ryanodin receptor had been studied by Wayne Hendrickson and Andy Marks for 9 years, and they never, with X-ray crystallography, and never got beyond 7 angstroms. And so with cryo-EM, we got first to 5 angstroms and then 3.6 angstroms within a very short time. Here's another collaboration that I am involved in at Columbia. The molecule, the receptor that is responsible for cystic fibrosis. Here's the publication, the first near-atomic structure that was obtained by at Rockefeller University. And here are our own contributions with John Hunt, with John Hunt's group, where we look at the relevant mutant that causes the disease. Okay, now from the work by Sriram Subramaniam, who pushed the resolution very high, we see that we can do a lot in order to get past the 3 angstrom limit And in fact, meanwhile, we are past the 2 angstrom limit. And you see more and more features of organic molecules that we only previously saw with X-ray crystallography. So we can really clearly see that cryo-EM is now on its way to become a very strong competitor to X-ray crystallography. And I should, of course, in the terrible times that we have now, I should also show a cryo-EM structure of the 2019 COVID spike. This was obtained most recently by McLennan's group in Austin, Texas. It was just published in Science. And so here you see how cryo-EM can rapidly address very important health challenges that we face today. You can see from the trajectories of PDB release structures, you can see that we will very soon outpace the depositions of X-ray and NMR because we are now here on an exponential trajectory with depositions, whereas the number of released structures per year has pretty much leveled for both X-ray and NMR. So here I have just a couple of slides for 2 new technologies that I've been particularly interested in. One is time-resolved cryo-EM. The objective here is to look at short-lived states that only last for 10 to 1,000 milliseconds. So they are inaccessible with the currently very slow process of specimen preparation. And they involve a microfluidic chip. The first devices using silicon were developed at RPI and the Wadsworth Center. And now we are experimenting with new chips with an organic polymer with PDMS, and we collaborate with the Columbia Engineering Department. So the idea here is that we want to study a reaction in detail. We have one component and 2 components. In the second component, we mix them for a very short time, for half a millisecond. And then comes a reaction chamber, which has different lengths for different chips. Here are the different lengths. And then in the end, with the help of some driving gas, the mixture of the various reaction products is sprayed onto the grid as the grid is plunged into the cryogen. And as an example of a biological process that we studied with this, I show the termination process in the ribosome. When a stop codon is encountered at the A site, then a release factor is supposed to bind. This is, in the bacterial case, either RF1 or RF2. So either RF1 or RF2 stop at the bind at the stop codon, and then they have to reach the peptidotransferase center to release the new peptide, not the new peptide, the new peptide. And then this whole process goes into the release process and recycling process. Okay, so we want to understand this. And the funny thing is here that both RF1 and RF2 are in a compact conformation. They are they're too compact to reach the peptidyl transferase center, which is 73 angstroms away. The compact structure is only 23 angstroms in size. So after the binding, one had to postulate that there was a conformational change, which then placed a domain 3, in the vicinity of the peptidyl transferase center. So in other words, there is a short-lived state in which the RF1 or RF2 are bound at the stop codon before they make a conformational change and then reach out to the peptidyl transferase center. Okay, so with the machine that I showed you, we have now reactant 1, which is the termination complex. Reactant 2 is RF1 or RF2. And so now we bind it, okay, and hooray, 24 millisecond, we actually see the compact state of RF1. It has not extended yet, so the experiment was successful here. This state has never been trapped before, so it shows that this whole technique can be used to trap something at 24 milliseconds. And we wait, now we do another experiment, and this time we wait for another 40 milliseconds, and then we see the extended formation. So now we know that between 24 and 60 milliseconds, this whole conformation change takes place. Okay, so the other technology, just a couple of slides, has to do with the mapping of continuum of states. Well, if we have very large data collections and we have this very large ensemble of images, and if you have such large ensembles, then even states of molecules that are very rarely visited become visible. So for a large enough data collection, we actually can see the continuum of conformations, but we don't have a tool in order to access this continuum. The existing tool is something like RelyOn, using maximum likelihood, which only find clusters in this continuum, but we want to really characterize the continuum. And the technique that I've been developing together with Abbas Urmaz in Wisconsin is doing this by so-called manifold embedding. So it's a way of flattening this distribution of conformations into a low-dimensional space, and then study this low-dimensional space for systematic changes of the structure. So we need to use some kind of an embedding technique, manifold embedding. Okay, so the first application was to the ribosome. It was in 2014. Yeast ribosomes purified from cell extract. No ligands were there because it was a relatively crude treatment. We wound up with 850,000 cryo-EM images. And at the time, with the technique, we could only use the great circle on the angular sphere that reduced the data to 80,000. It was published in PNAS in 2014. And so what we see is really, we could, from the distribution in the low dimensional space, we could then use a Boltzmann relationship in order to estimate the energy landscape. And here's the energy landscape of the ribosome, of the yeast ribosome. And there's a suggestion of a cyclic process. And we got very excited and showed and looked how it was related to what was already known about conformational changes of the ribosome in the elongation cycle. And indeed, it looked as if the ribosomes, even without ligands, actually perform all the different conformational motions that would be required if they were to do their work in the cell. So this is just gives you a glimpse of what can be done with this technique. So with this, I'd like to come to a conclusion and an outlook. Cryo-EM is a dynamic field which will have many surprises in the future. And single particle cryo-EM is a game changer in structural biology. And the ability to retrieve multiple conformation is a particular forte, promising to reveal the functional dynamics of a molecule. And time-resolved cryo-EM can give detailed kinetic information about a reaction of two-component system on the time scale of tens to hundreds of milliseconds. And the analysis of large cryo-EM datasets can provide information about the energy landscape of a molecular machine. So here, at this point, I would like to reiterate thanks to the many students that have been working with me over the decades of this development, too numerous to list, but I do want to list my collaborators. These are collaborators partially in the ribosome field, but also in the development of techniques. And then I'd like to give special thanks to my wife, Carol Sagenow, for 37 years of unwavering support. And Carol would be with me if we didn't have the pandemic, and we would be enjoying ourselves in Vienna and the Prater and all the good things that one can see in Vienna. Okay, and here I conclude with a picture that was a year ago on the top of the of the building where our lab is in. Thank you very much for your attention.
Thank you very much, Joachim, for this. I'll try to do virtual applause for this very comprehensive kind of historic overview. Really enjoyed it. And are you willing to answer a few questions?
Yeah, sure.
Yeah. So maybe I'll start with, apart from the avenues you kind of already explored and mentioned, what do you think are kind of challenges for cryo-EM in the near future? For example, do you think 100 kV microscope as proposed would become alternative? Or?
Yeah, I'm very much in favor of these less expensive versions of the microscopes, and there have been a number of studies now that show that 100 kV is really sufficient in many applications. And as we all know, the microscopes that are currently in use everywhere, the Krios Titan, is essentially unaffordable to a regular lab. And the national centers are only part of the solution, because very often you actually need to be, right by the microscope in order to interact and develop specimens further and obtain rapid feedback. So these big machines are to start at $5.5 million. And so, the 100 kV machines would really be a very big new development.
Thank you. And maybe like another kind of limit for cryo-EM right now is the small size proteins. What do you think can be done to get smaller and smaller?
Yeah, smaller and smaller. One is that I believe there's going to be more sensitive cameras that come out, and they reduce the noise even further. And so that shifts the, the molecular size down. And then a faceplate is another development which should reduce the noise and well, it doesn't reduce the noise, but it enhances the contrast at low spatial frequencies. So that is a development. The other one is it's very interesting. There has been a publication on this last year. The ability to attach smaller molecules to a large carrier molecule and then reconstruct both together. So it has been demonstrated by a group, and I can't remember the name of the group, that this is doable, and one can really achieve the same resolution as before if one uses the some kind of an particular tagging technique.
Yeah, thank you. I'll see if there are any questions coming up. Yeah, I already asked one, kind of one of them. I think one question was, what do you think is the challenge in using cryo-EM for small molecule drug discovery?
Yeah, it's really for drug discovery, you need to get into the right range of resolution. And most recent publications have shown that obstacles can really be removed, and one can get into a range of resolution that's better than 2 angstroms. And that's really between 2 angstroms, 1.5 angstroms, we really get in a range where we can use the existing methods of drug discovery. And this is a very exciting development. You know, in looking at the ribosome, we're very often happy to get too close to 3 angstroms, but that's obviously not enough for drug discovery.
Thank you. But I'll see if there is, if anybody wants to like raise their hand and ask a question, now is the time. I don't see any hands so far. Oh, no. Paul Shanda, it's coming in, kind of disappears for some reason. Yeah, coming in IST faculty, wants to ask a question, please.
I was curious about your view on hybrid methods, that is EM combined with cross-linking mass spec, with NMR, with this kind of approaches, biochemistry. So what can be done? Maybe can this be used to push resolution? And what do you think are the limits there? What are the opportunities?
I think you talked about cross-linking. I'm really I'm a bit nervous about cross-linking, about the changes, structural changes that can happen. But interdisciplinary approaches where you use multiple techniques are obviously give you many different aspects. And I'm not so familiar with what NMR can contribute here. Mass spec obviously is a very powerful additional technique. And what I'm particularly excited about is the interaction with fluorescent resonance spectroscopy with single molecule FRET.
Yeah. So
then we really see complementary aspects of the molecule. We obviously can do nothing in real time, and then it's very exciting to combine what we see structurally with real-time studies such as single molecule FRET.
Okay, thank you.
Let's see if there is any more. Okay, I don't see any hands. There is a question from Manjunath Javur. How do you approach solving a protein structure which is dynamic and also has no initial model available?
Do I understand it right? A protein structure that is dynamic?
Well, this you more or less addressed, but how do you generate initial models? The question is.
Oh, the initial models. That's very. That has been done in many times for completely unknown proteins, because there are techniques that are bootstrapped. They are different from what I showed, because they are new techniques that have been developed, in which you can just essentially take the shape of a potato, and in order to get the bootstrap reconstruction.
Mm-hmm. I have maybe a kind of related question. I mean, now we have at least 5 or more software suites to do single particle, at least, and each of them has its own advantages and disadvantages. Like, for example, from initial model generation, from CryoSpark, Spark migrated into Reliant, and so on. Do you see any way this can be kind of united into bigger suite? Is there any initiative for this?
Well, it's already been united. There are platforms that have been created for the specific purpose of letting these different softwares interact. And Scipion is one of them that was developed in Jose Maria's lab in Madrid. And Appion was an earlier version, not a version, but an earlier platform that was developed by Richard Carragher and Clint. So there are already platforms that encourage you to explore different flavors of software, and in order to get some kind of an optimum solution. And this is a very encouraging development, and I think I would encourage everybody to make use of this. And in my lab, people have to get sophisticated and look at different softwares, and they typically combine them just in an ad hoc way and not on platforms. But I very much encourage to use the existing platforms.
And your Spider now, you don't develop anymore?
No, that unfortunately, when I moved to Columbia and the Spider development was still in the hands of Dean Leith, I was very happy that I didn't have this kind of burden anymore, and I wanted to really focus on on ribosome structure and so forth. Then Dean retired, and so we didn't have a very good solution to maintain us with the current explosion of technology. So an entire group is really required to keep up with this. So we have now there's one version of Spider which has been maintained by Michael Rademacher in Vermont, in Burlington, but he's also retired now. So the caretaker is Tapu Shaikh, who I believe is now in Dortmund. And he was able to implement Spider on Scipion, on the Scipion platform. So there's a certain subset, a very useful subset of Spider, which can directly be used instead and together with other software systems.
But your recent developments on energy landscape, they will be kind of independently available software.
This is software which has been, developed in collaboration with the Wisconsin group, and that was a MATLAB development, which then got ported in Python, so that we could really handle it, and a GUI was created. It's a very sophisticated GUI, and we call it manifold EM or manifoldum. And so this is, we had an alpha group of people giving us feedback, and we will soon release it in a beta version. And so this all the software is really then can be controlled with a GUI, and you can run it in your own lab.
This is really great, because this is one of the features we really need for like big molecular machines. So we're looking forward to it. So I think we are now probably mostly out of time. So I want to thank again Joachim for the great talk, and yeah, we really appreciate it.
Okay.
Thank you very much.
Thanks for your attention and your interest.
Thank you very much.
Bye-bye.
Bye-bye, everybody.